For those that are just joining us, I'm Ethan Etzen, co-founder and CEO, and joined by Liam Welgengun, our senior mechanical engineer. And we'll jump right into our presentation. Our company is called Ocean Diagnostics. We are a Canadian environmental impact company. We're based on the west coast of Canada in Victoria and Vancouver, British Columbia. And we were founded in 2019 by engineers and marine scientists. And we are really working on trying to develop innovative technologies to overcome some of the challenges with various pollution and biodiversity issues. So as a company, we design underwater sampling equipment or in situ sampling equipment. And we also operate a service laboratory for microplastic pollution. So the focus of today's talk is going to be on eDNA and our eDNA surface sampler, but we also do work in other types of pollutants and contaminants as well as a company. We collaborate with governments, research institutes, private industry, a lot of different conservation organizations, First Nations and Indigenous groups, and environmental consulting firms on a regular basis. So as a company, we're focusing on two large global complex problems, and that is plastic pollution, specifically microplastic pollution, and changing biodiversity. So I think many of you on the call are scientists or work in the environmental field. You probably know a lot about these two issues that are facing our planet. Specifically, we focus on microplastics and trying to better quantify and analyze microplastics in the marine and freshwater environments. And that covers a wide range of different analytes and different ecosystems that we monitor. We've also been looking at sort of changing biodiversity and biodiversity loss and trying to find scalable techniques for making it easier to track and monitor biodiversity on an ongoing basis. And we'll talk today about one of those approaches, which is environmental DNA. So as a company, we focus on kind of three main areas. We design sampling technologies. In this photo, you see our depth sampler called Ascension. This is a device that can go down to 400 meters and collect in situ filtered sample and sensor data. We also have the surface sampler, which we'll talk about today. We offer a microplastics analytical service out of our lab in Vancouver. And then for groups that need a little bit more help, we also offer project management and consulting. So this involves study design, getting into the field with our technologies. We can do analysis of the samples and then helps groups to interpret the data and make sort of informed decision-making afterwards. So these are the three areas that we focus on, and we have products and services kind of throughout each of those. So what is eDNA? Environmental DNA is a fairly new approach in the grand scheme of science, I guess. It's been used kind of over the last maybe 20 years or so, but over the last 10 years has really matured as a technology. So all living organisms have and shed DNA into the environment, and environmental DNA is just DNA that's been released from an organism and is kind of free-floating or readily available in the environment. So this could be skin cells, hair, scales, waste products like feces or urine, or reproductive cells that come off of different animals as they interact with the environment. And environmental DNA remains stable in the environment for anywhere from 1 day to months, depending on the sort of conditions and UV exposure and temperature and things like that. And I think we're getting better at in the scientific community about understanding that residence time. But what it means is that we can, after the fact, sort of as a detective, take environmental samples and understand the organisms that have been in the environment previously. And this is a very powerful tool for biodiversity assessment. So what are some applications for eDNA and specifically eDNA surface sampling? Here's 4 that I think cover some of the areas that we see our partners working in the most. So the first is impact assessments for coastal infrastructure. So as a human population, we tend to build very close to the coast or close to waterways. This is very helpful for things like trade and access to food, things like that. So as we build and interact with the coastal ecosystem, it's very important that we perform impact assessments to understand how us building and interacting with the coast is affecting the local habitat around there. Ecosystem assessments, so think fisheries. As we get a lot of our primary protein as a human species from the ocean, and understanding the stock levels of fish or the migratory patterns of different organisms is quite important for understanding how the ecosystem is behaving and how healthy it is, and therefore how we can harvest food from the ocean sustainably. Invasive species tracking, this is a multi-trillion-dollar problem that affects many different marine and freshwater industries and areas. Pictured here is the zebra mussel, which is kind of a famous invasive species that has wreaked havoc on freshwater ecosystems in North America and elsewhere. So being able to quickly detect invasive species before they are allowed to inhabit and completely take over an area is a great, eDNA is a great sensitive technique to allow you to do that more easily and efficiently. And then, of course, monitoring species at risk. So there's many species that are on the IUCN endangered list, and our ability to kind of track these organisms is generally very hard, especially as populations start to dwindle. So using more sensitive molecular techniques like eDNA allows us to track the migratory patterns or sort of geospatial coverage of these organisms in the environment much more sensitively than we can just by deploying visual surveys or scuba diving down and looking for these organisms ourselves. The eDNA pipeline is quite complex, and there's many different analytical assays that can be used to understand what's happening in the environment. But here's two different things that are the most common. So it all starts with collection of an environmental sample. This could be a water sample or a soil sample or an air sample, for instance. DNA is extracted from that sample using a lysis buffer to basically extract and remove and purify the DNA from all of the other things that come with the environmental matrix. And then there's two main approaches that you can use for eDNA. The first is quantitative PCR or digital PCR, and this is a single species specific assay. And this allows you to create a target assay that's looking for, let's say, a rockfish here in British Columbia, a specific species of rockfish. We can design a primer that is looking just for the DNA of that fish, and then we can compare the DNA that we've extracted from the environment and create a quantitative or a digital readout of the amount of copies of that DNA that we find in our sample. So this is really useful for presence or absence of a single species, or trying to do semi-quantitative work of the amount of genetic material that's actually in your environmental sample. The other approach is metabarcoding, which is taking all of the DNA in your sample and using a bunch of what are called universal primers. And we can apply a series of these to basically understand the entire tree of life that comes from our environmental sample. So the DNA that's extracted is amplified to a level that we can read it. It's put through next-gen sequencing machines that read all of the DNA and sometimes RNA that is in the environmental sample. And then through a technique called bioinformatics, we can match those reference sequences up to reference databases that are available publicly. And we can get a better understanding of all the different organisms that are in that environmental sample. And this is very powerful for understanding overall biodiversity or species composition in an area, depending on what your target question is. So the first part of that is sampling. And at the end of the day, you need to get a water sample if you're doing water eDNA. And here's some of the most common water collection methods or water filtration methods that we see for environmental DNA. The first one is just basic syringe sampling. It involves taking a normal medical syringe and slowly pushing water through that syringe over a small membrane filter over and over again until you've basically saturated that filter. So this is generally several liters of water, something like that, to be able to get a good eDNA sample. The second is gravity filtration. You see here some sort of IV bags. These can be filled with environmental water, and then just the weight of that water can slowly be used with gravity to filter through the filters beneath. The next is setting up an entire filtration manifold. So this uses a vacuum pump, and you set up a series of these manifolds and put filters on and just pour the water directly over the filters as you're using a vacuum to pull pressure on the filters from behind. So these are generally more used in lab setups, but you can bring them out into the field as well. And then lastly is sort of in situ or field vacuum pumps. So pictured here is the Smithroot sampling backpack. Smithroot, if you guys don't know, sort of, I would say, pioneered the portable vacuum pump probably about 10 years ago or more. And this is their product, which is basically a Ghostbuster style backpack that you can bring out into the field to directly do this vacuum filtration in the field. And this really enables people, I think, to be able to collect samples more easily out in the field. So here's some of the techniques. Each of them have drawbacks. A lot of them are prone to contamination. So having to decontaminate or sterilize a lot of the glassware or the things that you're using in the field to make sure that you're not getting cross-contamination of DNA from one site to another. And that makes it very difficult to do this type of work. So looking at these available options, at the company, we tried to look at what was available in the market and see if we could help. And with in partnership with Dr. Peter Thielen at Johns Hopkins, who was working on a sort of even smaller, more portable pump, we conceptualized an idea to bring something to the market that would be really easy to use in the field. And what we ended up with after about a year and a half was the eDNA surface sampler. So we wanted something that was very small, could fit in your backpack, easy to take into the field, and provides a lot of the benefits of not needing to sterilize or decontaminate systems, and something that can provide enough suction pressure to actually do DNA filtration directly in the field. And this eliminates a lot of those challenges that we talked about before. All right, with that, I'm going to move over to Liam here, and we're going to go through sort of an unboxing and a product demo so you guys can see the product in real life. So Liam, I'll pass it over to you, and we'll go back and share our screen here.
Beautiful. Thanks, Ethan.
Just give me 1 second.
While Ethan is getting ready here. Oh, top-down camera already. Look at that.
Okay, I can switch to...
I think you're still sharing the screen there.
Give me the...
Oh, that's perfect.
Okay, great.
Yeah, thanks, Ethan. All right, guys. It is playtime. Here is the Ocean Diagnostics surface sampler. In summary, it's a suction pump. We elected to make the most purpose-built, field-ready, high-performance suction pump imaginable, or that we could imagine at least. It's an entirely wireless and portable design. And so to make it wireless and portable, we chose to power it with rugged, dependable, and globally available Makita 18-volt LXT batteries. They are hot swappable, quick charging, water resistant, and we've deemed them to be the best batteries on the market. They also include a battery management system, which is quite uncommon for these particular types of batteries. And so it protects them from a whole bunch of different scenarios that could potentially be damaging, whether that's in the field, in the lab, etc., which bolsters the reliability of the product. And so they very easily mount directly onto the surface sampler, as you can see here. And one thing I'd like to note at this time is that the battery is not included in the sale of the product. However, the protective case that we'll talk about a little bit later, as well as the tubing and some fittings, are. A little bit more on the battery. These guys are very globally available at short notice and in a variety of sizes. You can find them online or at your local hardware store. And one more thing to note is that the 5 amp hour version or less, you can legally take in your carry-on on the plane. So whether you're doing some sampling abroad, you can travel with this with ease, and you don't have to worry about it getting lost in your checked baggage. But anyways, yeah, these 5 amp hour batteries will give you about 5 hours of 100% duty cycle heavy load field operation, which is more than enough for most applications. But say you're doing a multi-day event out in the mountains, doing some remote field sampling, etc. You can always buy another one of these batteries and huck it in your backpack and yeah, hot swap them to do with your job. But anyways, what this battery powers is essentially the most powerful and compact self-priming diaphragm pump available. It does not need to be primed. It can run dry. And this is essentially lets you focus on your mission rather than a complicated SOP. So being a suction pump, the pump is furthest downstream in the sampling apparatus that you're using in the field, meaning it employs a filter-first approach. That means you don't need to sterilize the surface sampler in between samplings, and you can achieve continuous back-to-back sampling in the field. This is yet another consideration for the simplification of field operations, minimizing your time required in the field. Internally, we've used a parallel fluidic system, which gets you both high flow rate as well as high suction pressure. So you see flow rates of around 600 milliliters per minute at maximum, and at suction maximum, you'll see around negative 95 kilopascals or negative 13.8 psi, which is perfect for environmental DNA. So to interface with the surface sampler, you use these two quick connect fittings at the front of the surface sampler. They are auto sealing, auto locking, quick releasing via the press of a button. They're also made of tough polypropylene, which is UV resistant, impact resistant, etc. And they won't crumble away in a few years' time. They have a matching component, which mates with them directly, which comes included in the two 1 meter sections of high performance silicon tubing, and they easily connect into the surface sampler. And that's all you need to do. They're sealed and ready to go. Included in the rest of the product sale, there are a bunch of other fittings here. 1/4 inch, 1/8 barbs, quick connect fittings, as well as lure lock fittings. And essentially, this allows you to interface with universal filters, universal filter compatibility, whether you're using 47 millimeter filter housings, capsule filters, or essentially whatever's in your SOP. There's a very good chance that what we've included in this kit is what you need to plug and play this solution directly into your SOP, your standard operating procedure. So taking a quick look around the rest of the surface sampler here, one thing you'll see is a single button on it here. This is the power button to turn the surface sampler off and on. It is the only button on the surface sampler, and that is again for ease of use. The body here is made of both impact absorbing as well as highly textured material. So even with wet, cold, and uncomfortable field hands, you'll have no problem handling and operating the surface sampler with a single hand, gloved, ungloved, wet, or not. On the topic of ruggedness, the surface sampler, the tubing, the fluidic adapters, the battery, etc., all the other bits and bobs fit within this Nanook 905 rugged case here. It is waterproof, dustproof, and this brand here, Nanook, they market it as being indestructible. And so far throughout our years of using them, we believe it. Inside, we have some custom cut foam, which is both serves to organize all of your contents inside, as well as it helps protect against impacts, etc. Additionally, in the case, or mounted on the case here, you have a pressure release valve. And so while you are traveling internationally, or say you're gaining quite a bit of elevation, you won't experience vacuum lock in your case here. That will prevent you from accessing your kit. So you'll have continuous access. You don't need to worry about vacuum. All in, with the surface sampler, the case, the 5 amp hour battery, etc., the whole kit weighs just over 1 kilogram, which is again in the name of being extremely maneuverable and fieldable. So again, moving back to the surface sampler here, just to finish up a few other product features, one thing you'll notice on the bottom are 6 rubber feet. These guys are a medium durometer rubber, which allows the surface sampler to dampen its own vibrations. So whether you're using it in the lab environment or whether you're using it in other sensitive environments here, it won't transfer many vibrations to other instruments nearby. Additionally, they are very, very grabby. And so whether you're using it on angled surfaces, say on boats in non-ideal conditions, it will stay nice and planted. You will also see on the bottom here these little silver bits. I'm not sure if it shows up beautifully on camera, but they are threaded inserts into the bottom part here. And it's a standard bolt pattern, which allows you to mount any number of essentially brackets to it. You can include it in additional like apparatuses that you have pre-existing. so it can fit into your existing systems. And one example that we've printed ourselves in office actually and designed is this little GoPro mount here. An application for this guy could say, if you have a boat hook on your boat or a pole, you can directly mount the surface sampler to that to get it closer to the water or to sample a particular body. Or if you're interested in marketing is asking you for some comment, you could, I guess, mount a GoPro camera on the bottom of this guy. In any event, the bolt pattern here on the bottom is entirely up to your creativity in terms of what you would like to make or design for that. Yeah, anyways, that is a quick walk around the surface sampler here. We're going to jump right into a product demo. So first thing we're going to do is we're going to assemble the surface sampler by inserting the battery. Just slides right in, locks on, release button at the front. We are then going to connect our fluidic tubes here. And as I do this, one thing I would like to point out is that we have our both input and output indicated here on the front, as well as their flow arrows on the bottom as a backup. And I would like to reiterate at this point that this is a vacuum style pump. And so we will be sucking through the input, meaning that our filter will be upstream. So we are going to attach our fluidic adapters 1 and 2. And now I'm going to bring the ocean to us, if you don't mind, which will be in the form of this metal bucket. And then our volume measurement device will be this tub here. Different people use different volume measurement devices. Some people use Nalgene's, some people use these buckets, etc. You can use a scale if you'd like to measure the mass of the water. We are going to simplify things and use the bucket today. We are going to be using a 47 millimeter filter housing for our little sampling demonstration here. And in the included kit, we are going to be using one of the lure lock fittings that quickly adapts onto the filter housing. And so for the purposes of speed here and not wasting anybody's time, let's say that this filter housing has been purged already, which it is with a 0.45 micron MC filter. We will just directly put that into the water here. Battery is mounted. All the fluidic connectors are in place, so we can just hit the power button. And we are sampling. I'm not sure if the fluid flow shows up here on camera, but we are sampling quite rapidly here. One other thing I would like to point out at this time, hopefully I don't have a mess, is that the surface sampler button there, the power button, does light up when you turn it on. So that is yet another thing that we have included to simplify field operation in non-optimal environments. Just reaching 500 milliliters there, we're going to shut it off. Depending on what your SOP is, you'll filter for different volumes, obviously. And I would just like to show you an example of 47 millimeter MC filters that we have seen before, pumping in something relatively similar to this product. So this is what you can expect to see pumping our tap water here, actually, as an FYI. But anyways, say you're done sampling here. All you need to do is disconnect your filter housing, reattach another one, and you're good to keep on sampling in a different environment or the same environment, because again, everything is downstream of the surface sampler. So to pack it up, all you have to do is run it dry here so we don't build up mold in our case. And then when it's convenient for you, you'll do a freshwater rinse to clean the diaphragm pump inside. You don't need to bleach it, you don't need to sterilize it, etc. Just run it with clean water. Push the 2 buttons here to detach the fluidics. Remove the battery, and you're all set to go to pack it away neatly inside the case. Yeah, so we covered that the surface sampler here has a self-priming vacuum pump. It's one of the most high-performance ones on the market, particularly in terms of energy density for its size. Given the Makita batteries here globally available, it is incredibly shield portable and rugged Everything is splash resistant in the design as well. Yet again, that gives you that reliability and non-optimal condition use. It is incredibly deployable. The barrier to entry with this product is extremely low, so it's easy to teach your staff. Yeah, it uses the filter first protocol as well, so you can continuously back-to-back sample without needing to worry about contamination, lessening your time required in the field. It also has universal filter compatibility, like we were saying, given all of the additional fluidic adapters that we give you, as well as the SOP integration, whether you're integrating that into an existing apparatus or an existing standard operating procedure in the field. It'll just decrease your complexity. And yeah, again, it's a low barrier entry to use, a dead simple design with the single power button, etc. The simple batteries, cordless batteries that everybody is familiar with. And yeah, thank you very much for listening to the features and the product demonstration here. Feel free to download the spec sheet and ask us any questions at the end.
Great, thanks, Liam. So after the product demo, I thought it might be useful to look at some of the performance in different types of environments. Obviously, the performance is going to vary greatly depending on where it is that you're sampling. So like in an estuary or a turbid river or in the coastal ocean or in blue ocean, as well as the pore size and the actual physical size of the filter that you're going to use. So we have a couple performance plots looking at estuary and coastal water here in Victoria that we collected with a variety of different filters that may be common to you. So on the plot here, you'll see on the y-axis we have volume, and on the x-axis is time. So this is basically just a representation of how much time it takes to collect a 2-liter sample, which is kind of a standard DNA volume that we find. And so looking at different filter types, you'll see that basically the larger the pore size filter and the larger the diameter of the filter, the faster your pumping time will be. So for a 5 or 1.2 micron MCE filter that is 47 millimeters in diameter, like what we just showed in the demo, fitting in one of those housings, generally we can collect the 2-liter sample in between 5 and 5.5 minutes, something like that. And then moving down to 0.45 MCE 47 millimeter filters or Steravex, that time basically doubles. And this is specifically in an estuary, right? So this is a pretty highly turbid, sort of tidally flushed inlet here that goes through downtown Victoria and into an estuary kind of place. So this is, I wouldn't say worst case condition because there's probably more turbid rivers than this, but this is quite a turbid area that we were sampling in. Looking at the same plot in coastal seawater, we ran the exact same tests just offshore. So this is in Oak Bay, Victoria, which is direct access to the Pacific Ocean, just off the coast of where we live here in BC. What we see is that not much of a difference in the 5 micron or 1.2 micron 47 millimeter filters, generally about 5 minutes or so. But we do see much better results with the 0.45 MCE and the Steravex filters. So going from 11 or 12 minutes down to 7 or 8 minutes or so to collect that same 2-liter sample in coastal seawater. So filter compatibility, I think, is probably one of the biggest questions that we get. And what we've found is that specifically for vacuum filtration, you're going to have the best success when you have more surface area or kind of generally larger pore size filters if you're trying to saturate that filter. So on the left, you'll see some of the smaller filters, so like the 0.45 Steravex or the small capsule style syringe filters, like 25 millimeter or even the 13 millimeter filters. We don't find the best performance of these, to be quite honest, with these types of filters, just because we're applying pressure from the back of the filter and the actual filter area is so small, it can be a little bit difficult, or basically it can just take a really long time for you to collect that 2-liter sample. We found much better success using 47 millimeter filters, so not 25 or 30, but 47, which has more surface area and is just going to give basically more reaction sites for that water to flow through different pores. And that goes with like 0.45 or 1.2 micron filters. Using the larger 47 millimeter filters, like the ones we showed in the demo, or Just another shout out to the Smith Root self-preserving filters that you see here. These are 47 millimeter style filters as well. Those tend to work really well with the surface sampler. I'm also learning about new filters all the time. And the top left is the kind of larger Nature filter. And then the top red is a new filter we just got our hands on called Silphium, which is sort of a sandwich style filter that has two rectangular membranes on the top and bottom. And it gives you a ton of surface area. I think it's about 2 or 3 times the amount of surface area that you get from a 47 millimeter filter. So if you're in a highly turbid environment, moving to some of these kind of larger pore size filter options is going to yield the best results with our surface sampler. I think a little bit of R&D needs to happen, I guess, when you first get it, but just wanted to have this slide up just to show, where we've had the most success. So after looking at performance, now let's get into some case studies. You might be wondering, where is this being used right now, and what are some different case studies, and how are people using the sampler to collect eDNA? So we've got 3 different case studies that we're going to present. The first is looking at kind of active fisheries management in the Pacific Northwest of America. The second one is looking at tracking humpback whales here in Victoria. And the third is looking at detection of invasive species, largemouth bass, in Quebec. So the first case study is with our partner, John Hagan, down at the Northwest Indian Fisheries Commission in partnership with WilderLab, based out of New Zealand. The Northwest Indian Fisheries Commission is a resource for 20 different indigenous tribes that are situated on the Olympic Peninsula. And all of these tribes have individual monitoring of their own fisheries. So what this group does is they are a resource to these groups to look at salmon habitat primarily, but all different types of fisheries habitat. And what this means is that they need to hike up into really, really remote tributaries and streams in the Olympic Peninsula to understand where these salmonid species are living and going to spawn. So that means that they're hiking really, really long, grueling days up into the mountains. And having a small compact kit has been a really great thing for them to be able to collect these eDNA samples in the field. So John's group has been doing some great work. They have 1,300 plus freshwater sites across the Olympic Peninsula. And the data that they're collecting with eDNA is directly informing fisheries conservation with these groups. And one recent finding that we just talked to John about was the detection of Chinook salmon habitat in a few of the headwaters that previously they had assumed were completely void of the species. So this is important from a restoration and a conservation perspective. With limited capacity, if these groups are going to be working on restoring and conserving specific tributaries, if they're interested in keeping the Chinook salmon population happy, it's really great to know exactly which streams they're going to on a yearly or every 2 years, so that they can focus their restoration efforts there and make sure that they're clearing debris and leaving plenty of room for those fish to get upstream to spawn. So for John, it's really great to be able to have such a small sampler that he can bring up into the mountains in these really remote streams. And we're really proud to be working with him. The next case study is looking at humpback whale monitoring here in Victoria. So we're working with PhD student Amy McNult in Karen Helbing's lab at the University of Victoria. And their group is looking at whale genetics primarily. And typically what they've normally had to do to get DNA from whales is take a Zodiac like the one you see in the picture on the left and literally drive up alongside of a humpback whale and take a full tissue biopsy sample using a tissue biopsy pole that actually removes a little bit of skin from that whale. And as you can imagine, this is not only very difficult to obtain, but it can be distressing to the marine mammals. And so their lab, which is specializing in all types of molecular techniques, has been using eDNA as a new tool to get that same genetic information without needing to do the tissue biopsies. So what they're doing instead is following these whales from a safe distance, with the permits that they have, and they're collecting eDNA data directly behind the whales. And having a compact sampler like the surface sampler that they can set up on a Zodiac that is, crashing around in the waves in a very small space allows them to be able to continually be filtering eDNA out in the field for an entire day. And this has been a real game changer for these guys in order to be able to collect those samples a lot easier. So what they're doing now is comparing the eDNA data that they're collecting from behind the whale with the actual tissue biopsy samples that they have from known individuals, and they're expanding the genetic data set of previously unstudied whale data here in BC waters. So again, very happy to be collaborating with them and the amazing work they're doing here in Victoria. Our last case study is with the Canadian Nature Museum, which is based in Ottawa. And this is a group that primarily works across the province, but is working a lot in Gatineau Park, which is a federal park within the province. And within that park, there are 50 very remote lakes that are highly susceptible to invasive species. Specifically, the species they're looking at is the largemouth bass, which many of you are probably familiar with in kind of freshwater systems. So in these types of systems, there's a mussel, the eastern elliptio mussel, that is really important for nutrient cycling and water quality in these lakes. And the juvenile stage of these mussels has a life stage that actually lives on the gills of the common shiner and golden shiner, which are small minnows that are directly predated on by largemouth bass. So what they're looking at is with the proliferation of largemouth bass in these lakes, there's predation on the minnows, and that could be potentially hurting these mussel populations. So what this group is doing now is updating a previous data set that they have on biodiversity in all 50 of these lakes, and having a small sampler that they can bring out into the field is allowing them to create a comprehensive biodiversity inventory of the different lakes in the park, which is something that was just too difficult to do in the past. And they're comparing that data with some of the traditional techniques that they've been using in the past, like just going fishing or using fish nets and minnow traps in order to collect this species diversity information. So also with these elusive minnows, they're quite difficult to detect. And so having molecular techniques, which are much more sensitive, is allowing them really to understand the spread of these largemouth bass and how it might be affecting the minnow and mussel populations within the lakes. So again, really happy to be working with these guys, and love to support their work. Having a sampler that they can walk into these remote lakes with has been really helpful for them to collect the data that they need. Okay, so we wanted to leave plenty of time in this webinar for questions. We've got the demo here, but we're also happy to answer any questions either in the Q&A or in the chat directly. So please put your questions there, and we'll go through one by one. Thank you so much for your time, and we look forward to answering some questions.